So again, I'm back to bother you with more information. But after making my physics teacher and my engineering professor proud, I think I would be able to make my school chemistry school teacher proud also. So the next Nobel Prize, which is on the agenda, is the Chemistry Nobel Prize. So again, I'll start with the names. Caroline R. Bertozzi, Morton Meldal, K. Barry Sharpless for their work in click chemistry, in which molecules snap together fast and firmly without the need for a long, complicated process and too many unwanted byproducts. Their work has applications in the field of medical science, including the treatment of cancer. So basically, there is a lot of burden of communicable diseases due to lifestyles or pollution, environment, climate change, and whatnot. Many of these studies, there's not much surprise in it that they are focused on the disease part. So in this, there is a new term known as click chemistry. So as we move on, I'll try to explain as much as possible. So currently, in simple terms, what each of the Nobel laureates did, the work they did. First, Sharpless, he came up with the term click chemistry and worked exclusively on it. Then Meldal, he came up with a special chemical structure called triazole, which has many significant applications. Then Bertozzi, one of the main constituents, she took the next step of developing click reactions that could work inside living organisms. So she developed a term, coined a term known as bio-orthogonal reactions. They are a type of reactions which take place between or amongst biochemicals which take place among the living cells in living system without interfering with native biochemical processes. So normally we are talking about non-communicable diseases, particularly tumor and cancer. So what happens is there are some the cells of the body or the biomolecules of the body. Sometimes what happens is that the immune system or some chemicals will come across a term known as glycan. So they don't allow the immune system to fight off that tumor or cancer. So this click chemistry, normally when we try to devise a solution or some solution or some medicine for a particular ailment, we try a lot of combinations. We try some reactions which are very difficult to perform. We use catalysts, for example copper, to perform some reactions which have a lot of side effects, unwanted side effects, and which can be toxic to the body. So instead of that, what click chemistry, in simple words, if we have two Lego blocks, so they can simply click with each other. Or if we have a buckle strap, if we consider a seat belt, so there is a click sound, so they can be easily attached to each other. So what the basic premise is, instead of trying to force two different components, elements, compounds, molecules with each other, we can start with some firmly constituted or basic molecular structures which can easily combine with each other. One such compound is known as triazole, which is a combination of an azide and alkyne. So basically, I'll be dealing some chemical structures with it. So basically, this is what the molecule looks like, and that is the triazole. So it consists of carbon atoms also, and it consists of nitrogen atoms also. Nitrogen atoms we have from azide. If I go into the chemical formula, because that also excites me a bit. So there are normally three in an azide. Azide is a sort of a suffix or attachment. So here we have double bond, double bond. If you have idea of bonds, we have single and double bonds. So here it has a positive charge. So this part, this whole part, this bond can be connected in term to some alcoholic group or some carbon atom or any other, or sodium. So what this is the azide group. So here you can see there are three nitrogen atoms. They are double bonded with each other with this is the structure of azide. And alkyne, basically, if you consider, that is two carbon atoms basically in a triple bond with each other. We have single, double and triple bonds. So normally we perform a cycloaddition reaction. We have a cycloalkyne. So basically, if you have come across this type of hexagonal structure, so this is basically carbon atoms at the center of this structure, and they are further attached to, for example, hydrogen. So two, it has four valence electrons, and then each carbon can be other. Here, if we consider the triple bond, that is for alkyne. So this type of alkyne structure will be combined with this azide structure to give rise to what is known as a triazole, and which has the application in click chemistry and helpful. We'll study in detail also how it works, basic structure. So this is what is the entire thing is all about. So a little detail for you, if you're still some text I've also. So it is all about a particular type of reaction that can join molecules together quickly and predictably, just like a fastener, belt fastener, or just like Lego blocks, and which is so precise it can even be used to perform chemical reactions safely inside the living cells. So it is called click chemistry, concept that describes reactions which are ideal, highly selective, high yielding, and that work with many different types of substrates. Then the reactions are generally able to be carried out in the presence of other competitor molecules and even take place in water. Now, if you remember, if you have studied in lower classes, that water is a universal solvent. So normally whatever reactions take place, water is normally it is involved in those reactions. So those reactions, if they have to take place with precision to make the adequate product which we want, to make the ideal reaction, so click chemistry and Ms. Bertozzi gave the term bio-orthogonal reactions. So this is that concept, or this is the work which got them the Nobel Prize in chemistry. Then bio-orthogonal chemistry is a field in which click reactions are used to carry out chemical transformations inside biological cells. Technique offers a new way to study the natural processes taking place in cells, and might even provide new ways to deliver drug molecules to specific cells with increased level of precision. So what did the scientists work on? Chemists often try to recreate complex chemical molecules. So if they want to block pathogens in cells, even if in an effort, in a sincere effort, or in a vivid effort, ardent effort to find the solution to a problem, for example, a malaria vaccine or COVID vaccine, they try to force the molecules or the compound into one another. But with click chemistry, it is all going to be in the behind. Then instead of trying to wrangle reluctant carbon atoms, Barry Sharpless encouraged his colleagues to start with smaller molecules that already have a complete carbon frame, such as an alkyne. Then chemists choose simple reactions where there is a strong intrinsic drive for molecules to bond together. They avoid many of the side reactions with a minimal loss of material. Sharpless called this robust method click chemistry. So this is just some text, and Mendel through his experiments came up with a useful chemical structure called triazoles, which are stable and are found in pharmaceuticals, dyes, and even agricultural chemicals. So when we talk about biomolecules, it is for sure that I'm going to talk about carbohydrates also, because carbohydrates are one of the main components. Basically, we try to define the carbohydrates. They are sources of energy, and normally when we eat bread, potatoes, pasta, sugar, so all these are carbohydrates. And if we try to define them in chemical form, so basically the carbohydrates have this structure, CmH2O. So normally the hydrogen and oxygen are in the ratio 2:1, but that always does not stand true in some of the stoichiometric constituents. So basically, when we talk about carbohydrates, you might have come across the term saccharides. So these are the basic constituents which conform to the carbohydrates. And when we talk about saccharides, you might have come across various terms like monosaccharides, polysaccharides, disaccharides. So monosaccharide is a type of carbohydrate which cannot be broken down into simpler form. A very common example is glucose. So the glucose looks like this. This is another carbohydrates by the name of fructose, and this is another carbohydrate by the name of galactose. So what we need to understand it, another example which we come across. So if we consider the polysaccharides, monosaccharides are normally known as sugars. They are the simplest type of carbohydrates. And when we talk about important examples of polysaccharides in which multiple monosaccharides are bonded together to form a sort of polymer. So polysaccharides examples are starch, which is the main source of energy in plants, and glycogen, which is the main source of energy in animals. Then we also know about cellulose, which is a strong material which is found in the cell wall of the plant cells. And we also come across chitin, which you might have studied in biology also. It is the outer structure which gives a firm structure to arthropods. So this is just a little additional interesting information. So basically carbohydrates look somewhat like this. Basically these gray atoms, they correspond to the carbon atoms, and this red balls normally to the hydrogen or other elements like phosphor or nitrogen. So these are the components. So now we come back to the main study, that Bertozzi, using the work of Sharpless and Mendel, came up with an efficient, innovative method to map glycans, which are carbohydrates based polymers made by all living organisms. So now, when you know about the carbohydrates, it might be a little simpler for you to understand these statements. That is why I explained this carbohydrates. Our studies have led to the insight that some glycans appear to protect tumors from the body's immune system as they make the immune system. This I have shut down. This I have already discussed, so you can easily correlate to what I have said earlier. To block this protective mechanism, Bertozzi, they created a new type of biological pharmaceutical, and they have joined glycan specific antibody to break down the glycans. We'll discuss in detail the diagram and the procedure. And this pharmaceutical is now being tested in clinical trials on people with advanced cancer. So if I go back to school again, then make you visit school. So this is the basic structure of the cell. We have the nucleus and the endoplasmic reticulum, microvilli, cytoplasm, Golgi apparatus, mitochondria, and the energy house, the powerhouse of the cell, ribosome, lysosome, plasma membrane. So so this is what the cell, what happens in a tumor or cancer is this cell, a particular cell, it cedes to perform its normal function. And when those cells, when those affected cells, when those dilapided cells, they multiply, and then the cancer or the tumor spreads. So while most chemists perform their experiments in carefully controlled conditions, these bio-orthogonal reactions, building upon the concept of click chemistry, these are being developed by these Nobel laureates. So in a biochemical soup of a cell in which the particular reactions can take place with precision in an ideal environment. So I was talking about one of the toxins, the copper toxin, which is often found during the biochemical reactions. So now if we study the progress, so Bartosi's term developed new click reactions, including copper-free versions that avoid toxic side effects and build upon click chemistry. Then these techniques are allowing researchers to study aspects of biochemistry involving sugars attached to proteins. And this work offers new understanding of biochemical process. For example, molecular mechanisms that explains how viruses infect cells. So I think till here it is clear. Then how does it work? So I was talking about that click example. Basically, obviously the molecule will not look like this, not like a belt strap or this fastener. So this is basically corresponding to one end of the clasp is corresponding to an azide, which I have discussed. The other end is alkyne. This is just a sort of prefix or suffix, and this is attached to a molecule, a sugar molecule. Then this sugar molecule, by the concept of click chemistry, these are molecules which can interact easily with each other, and with the assistance of copper as catalyst, this forms the triazole, which we have already discussed. There were 3 nitrogen atoms and 2 carbon atoms, and further these molecules are connected. So this is a type of click reaction. Then click chemistry could be applied to many different chemical reactions. This copper catalyzed cycloaddition is probably the most important example. In this process, molecules containing highly reactive azide group are reacted with alkynes in presence of copper as catalyst. The azide and alkyne click together to form a triazole group. Now, how it is further applied to map the glycans, which Bartosi talked about, that was inhibiting the function of the immune system to cure the cancerous cells. So basically, I hope this diagram, if not visible, so basically we have already discussed this cell. This is a cell, it contains the nucleus. So Bartosi fed the cells with a modified sugar with an azide on it. This is type of a regulating molecule, regulating, there is a molecular handle as mentioned. Then this is the glycan, which is purportedly affecting the immune system. It is not allowing the immune system to battle the cancer causing organisms. So the modified sugar was incorporated into a glycan. It was incorporated, it has been sort of attached, and special carbohydrates located. Then this is a fluorescent green molecule. For example, when I'll come to the bottom image also. So this alkyne, and when this azide, glycan attached with azide, these are fixed together, then it forms a click reaction as we have discussed, and it forms a triazole attached to the glycan. Then Bartosi used an alkyne clicked with the azide, a fluorescent green molecule, type of molecule which allows the microscopic or observation of the glycan molecule and how it is reacting or affecting the cell. So fluorescent green molecule sat on the ring shade molecule. This allowed Bartosi to track the glycans on the cell's surface. So this is a cell membrane, this is the triazole, the fluorescent molecule, this is the azide connected to the glycan, and in this way the whole sugar molecule and how the glycan is interacting with that cell, and further how this method can be applied to finding solutions or to find medicines for the diseases, various diseases. This is what the entire work is all about. So this is the important work for which the 3 Nobel laureates were awarded. So this is just the description which you can easily pause the slide and mention. This is an alkyne structure. Then studying glycans, proteins, biological cells, Bartosi wanted to attach fluorescent molecules, and bioorthogonal chemistry. Then early efforts used involve the azides, and slowly and gradually we are building upon that. So this molecule under the microscope, it might not always be, if this is a cell and we want to observe, these are very tiny molecules. Just we were like we were talking about the quantum physics, these are not observable by a naked eye. We need to use a microscope, and even while using the microscope, since so many elements are, so many molecules, atoms are at work with each other, so we need some sort of indication, some sort of molecule which can be easily visible under the microscope. So this is an example of this highlighted part, this glowing part, which is used as a fluorescent molecule. So this brings us to the end of the chemistry Nobel discussion. I hope this was also informative to you. I would like to apologize to my chemistry teacher if I went wrong with some of the concepts, but I think I have done more than my best. So in the next video, we'll be talking about the economics Nobel. Stay tuned.